Genetic engineering rivets attention, and this is because it has found multiple applications. Now, the base or the foundation of this genetic engineering or modern genetics lies in a simple workflow, which is that of gene cloning. So, in this session, we are going to look at the general workflow of a cloning experiment. So let us look at the learning outcomes of the session. Gene cloning is a process which enables one to get multiple copies of a single gene, and these are referred to as clones. The workflow of gene cloning is generally of four main steps that is aided by several molecules in the form of biological tools. The four steps include cutting the DNA into smaller fragments, joining these fragments into suitable vectors, and these suitable vectors are then introduced into suitable hosts, and these hosts are then allowed to be cultured and grown. So the grown cultures will basically amplify that single gene that was originally introduced into the vector, and that is why it is called as gene cloning. Now, these recombinants are further used for various applications. So, when one asks what is gene cloning, the very simple explanation to this is that if you have a fragment of DNA which may comprise a gene, and if this fragment of DNA is multiplied into several copies, and each of these copies are identical to each other, then these are what are called as clones. So it is very clear that there is amplification of a specific fragment of DNA or a gene. Now suppose you consider that this is the gene for insulin. Then having n number of insulin genes will give rise to n number of insulin. So you will get a large amount of insulin in vitro, and that is important because insulin is required for treatment of diabetes and there is a demand for it. So through gene cloning, one can get a large amount of insulin as a product. The second reason why gene cloning has been carried out and at a very fast rate is because through gene cloning one has been able to get sufficient DNA for sequencing. So the sequencing of the DNA has enabled one to understand a lot about the genes and the gene functions. Therefore, the understanding the function and the structural aspects of a gene has been helped by the gene cloning. Now, these are just basic, uh, applications. But now, from this on, there are many applications that are related to these basic applications. So, the step one of gene cloning is to isolate the gene. Now, suppose you consider you have a long linear DNA that has been obtained or purified from either a bacteria or a plant or an animal cell, or one has a bacterial closed circular DNA, okay, then this DNA would have several genes within it. Now, say for example, one is interested in just say for example this gene and one needs multiple copies of this gene. Then how does one go about only getting this gene from a DNA which has several different genes? Same is the case for a circular DNA. How would say one get this gene from a DNA which has multiple number of genes? So the main thing with this is that you may get this gene if you just are able to get this portion, which means that you can try to cut the DNA, okay, such that you get fragments of DNA, and every fragment of DNA may comprise at least one gene. So therefore, now the task is that from these fragments, you identify that one fragment that contains your gene, or you clone each of them and then try to find out from the family of clones which one is the clone that is having the gene of your interest. In either ways, what is going to happen is you will get multiple copies of the same gene. You will get multiple copies of the same gene. So all of these are going to be identical, and that is why they are called as clones. Now, there are certain questions that come up due to this. Can one clone all the genes? The answer to that is yes. One can clone all the genes present in a single DNA. How will one identify the gene that is to be cloned? So, therefore, there has to be a strategy or there has to be a methodology to identify that from all the clones of a single DNA, which is the clone or which is the clone that is the gene of your interest. And also, how is this amplification done? How from a single fragment of DNA you are getting multiple fragments and all of them identical. So this is also something that has to be understood and is being done through gene cloning. Therefore, the step 1 of gene cloning is basically cutting of the DNA. Now, cutting, therefore, is definitely manipulating because you have a linear DNA and when you cut it into fragments, you are engineering the DNA. Now, let us go to step 2 of gene cloning. So, in the step 2 of the gene cloning, the fragmented DNA that is there is inserted into a suitable vector. So, that is the second step. Now, a vector is generally a closed circular DNA, whether it is plasmid DNA or whether it is a phage DNA. Most cloning vectors are circular and closed. So, in order to insert the fragment of DNA into the vector needs to be cut. So, when the vector is cut over here, you have the two free ends available and this fragment of DNA will have to get inserted into this vector at the cut region. In other words, what we have to understand is that the fragment of DNA has to be inserted and then joined because the ends of the two, that is, ends of the vector and the ends of the fragment of DNA, have to be sealed or completely joined. Now, this is possible when the fragment of DNA and the cut ends of the vector fit into each other, just like a jigsaw puzzle. Every piece fits into the other piece because of its specific, um, you know, a specific way or a specific conformation or a specific interaction. So, therefore, this is important, and that's the reason why a suitable vector has to be chosen. Now, this cutting of the vector is aided by a Caesar. So, one calls it a molecular Caesar because it is a molecule that is used to cut. And basically, Caesars or the molecular Caesars are doing nothing but breaking the phosphodiester bond. While if you look at joining, joining means you're sealing by forming a phosphodiester bond. So these two are opposites of each other, but unless you cut, the fragment of DNA doesn't get in, and if the fragment of the DNA gets in once, then it has to be joined. So therefore, cutting and ligation, so cutting and joining are essential part of gene cloning. So step 1 basically involved cutting. Step 2 is involving joining as well. So therefore these two mechanisms are part and parcel of a gene cloning experiment. Now the question that comes up next is why do we actually need a vector? So what is our objective? The objective is that you get multiple copies of this fragment of DNA. So, when you insert the fragment of DNA into the vector is basically a vehicle that will carry the fragment of DNA into a host cell. And when that host cell multiplies, you will, along with the multiplication of the host cell, have the multiplication of the fragment of DNA as well. So, therefore, a vector is needed to deliver the DNA into the recipient host. Number one. Secondly, the vectors are generally found to be autonomously replicative. So they themselves can replicate to form copies. And so you would not have just the vector having number of copies, but when these copies are inserted into different host cells and the host cells grow in the culture, then there is a hundredfold increase uh, in the multiple or in the cloning or in the number of the fragments of DNA. So to multiply into copies is the second important thing that the vector carries out. So these two things make the vector an essential in a gene cloning experiment. It also helps in manipulating the genes if need be for a specific purpose. And interestingly, if this vector is such that it also not allows the expression of the fragment of DNA, then you may also get a product. So you may not just only get multiple copies of the fragment of DNA, but the multiple copies of the fragment of DNA can also be expressed and you can get its product. So that is why the vector is an important and essential part of the gene cloning mechanism. Now let us go to the step 3 and 4 of gene cloning. So once you have the fragment of DNA inserted within the vector, then this entire molecule with its integrated fragment of DNA is called as a recombinant DNA molecule. So when this recombinant DNA molecule is transported into a host cell, then that process is what is called as horizontal gene transfer, which means that you have from the surrounding a, a DNA molecule entering a host cell, and that is why it is called as horizontal gene transfer. Now, one can carry out horizontal gene transfer by three mechanisms. One is what is called as transformation, second is what is called as transduction, and the third is what is called as conjugation. So transduction is specifically when you use viruses as vectors, while transformation and conjugation can be carried out for plasmid DNA. So this was step 3. So in step 3, what you carry out is transport the recombinant DNA into the host cell. That is step 3. Once you have the host cell containing the vector inside the host cell multiplies. Why? Because it is an autonomously replicative system and it can use the host replicative machinery to multiply. But this is definitely several fold increase in the number of the recombinant DNA. But when this bacteria further multiplies into 2, you can see that each of the bacteria that is formed will again have multiple copies of the same. So if you get 10 such bacteria, you can see how much more the clones have been formed. So therefore, that is basically what one understands as amplification. And so you will find that in a media, you will have bacterial colonies formed, and each of this bacterial colony is basically one clone. Multiple cells present within the colony, all of the cells contain that one single recombinant molecule. So therefore, each of the different clones can have, each of the different colonies can have a different clone. So this is basically what is the final step of gene cloning, and this is what is going to give rise to multiple copies of that gene of interest. So to summarize again the entire mechanism, let us understand that you have vectors into which the DNA fragments that are to be cloned are inserted, and then these inserts now carry different fragments within them. These inserts can now be transferred into a bacteria, and these bacteria can be plated out into a fresh media, and when it is plated out into the fresh media, each cell is going to form a colony, and every colony has thousands of cells present, thousands of bacteria present. So you can imagine that every colony has multiple clones of a single fragment. And that is how you have gene cloning happening. So let us make the conclusions. Gene cloning involves isolating a gene or fragment of DNA containing the gene, which needs to be multiplied or amplified. To enable amplification or multiplication, the gene is inserted into a vector which can autonomously replicate. For further amplification, the vector with the gene or the fragment is introduced into a suitable host organism, and this host with the recombinant DNA will be allowed to multiply in fresh media to get multiple copies of the same. Therefore, biological tools to cut, join, insert, multiply DNA, all of these work in tandem to get a gene of interest in multiples, and this workflow has led to the field of recombinant DNA technology and modern genetics. Thank you.